today. This is Greg Winter, a very good colleague in Trinity College. You know, he's going to be admitted, I can't remember the word, as Master
Installed.
Installed. In exactly 2 weeks' time, he has to come and on the great gate, which will be opened by the head porter, and all the fellows will be assembled by chance on the inside of the gate and ready to welcome him. So this is a great medieval process we go through on these rare occasions. But anyway, Greg Winter is a great molecular biologist and also a great entrepreneur. He set up a company, Cambridge Antibody Technology, in 1989. So he has great experience in the business side of protein technology. That was a company that, according to Wikipedia, I don't know if it got it right, it was acquired by AstraZeneca in 2006 for £702 million. So it did rather well. And since then, Greg has very recently set up a company called Bicycle Therapeutics, very appropriate for Cambridge. But perhaps you'll tell us a little bit about it. So, Greg Winter.
Thank you very much. So, Keith, thanks for inviting me. And a few nights ago, actually, Michael Berridge told me about his theory, and I was stunned and rushed out the next day and got myself some vitamin D and continued to be guzzling the stuff. So, in fact, I completely understand if, in fact, you're thinking about what he said, because he's managed to combine Alzheimer's and memory and sleep and, in fact, pretty much everything under calcium. And definitely worth talking about afterwards, because I think there are many things that bear thinking about. If you can focus for a few minutes back onto protein folding. I have become aware that mathematicians are very interested in protein folding, and I guess believe that this could be very useful one day. And what I'd like to do is to tell you, given that we don't understand protein folding really in great detail, then how could one, or how does one, go about making therapeutic drugs that are based on proteins, where we have to engineer them, but without actually being able to calculate in the way that a modern engineer would. So in some senses, we're in a kind of medieval phase as biologists, doing things as engineers, but I would like to point out that there are one or two mathematical principles involved, but not many. Now, I'll start off with the central dogma. Essentially, the code in the DNA makes its way through to RNA and into protein. And that's the same pretty much in humans and mice and bacteria or yeasts. So the entire living flora and fauna of the planet is based on the same code. The consequence of that is that if you were to take a gene, for example, from a human, and you were to clone it, that essentially means connecting it to a self-replicating piece of DNA, and put that into a host cell, for example, a bacterium, you can produce the corresponding protein in the bacterium. So we just say we're expressing the protein. It just means taking that human gene and putting it in a bacterium. The other consequence of this is if you choose to alter that gene and then you express that, you can then create the altered protein in that cell in the bacterium. And that is the way by which proteins can be engineered, essentially by tailoring the gene and putting those instructions into a host cell so that it generates the altered protein. And in fact, this became big business in the 1970s, shortly after cloning was invented, and in the, in fact, 1990s, and I'll tell you why in a minute. But Sidney Brenner coined a new doctrine, so DNA makes RNA makes protein. Sidney Brenner said, makes money. So basically, DNA makes RNA makes protein makes money. And the money came really from taking those natural genes, human genes, particularly things like insulin, human growth hormone, or erythropoietin. These are things which are produced in very low quantities in humans, and producing them in some host strain, which might, for example, be bacteria, or more usually now Chinese hamster ovary cells. The second wave of money making came really from the generation of altering proteins, engineered proteins. So essentially where genes had been altered by using synthetic or in fact recombination of natural genes. And in fact, the main embodiment of this has been in the antibody field. And so I'm largely going to focus on antibodies because I think they illustrate the important things, the kind of general principles you would use to create novel proteins. So if we look at the action of antibodies in nature, you're probably aware that infectious diseases provoke the production of antibodies, our sort of immune defense mechanism. And the antibody is a large molecule. It has a modular type of structure. It actually consists of 2 chains. In fact, it's 4 chains in total. 2 called heavy chains, which I've marked in red, and 2 light chains, which are marked in white. So it has this kind of Y-shaped structure. And the antibody exerts its therapeutic action by really 3 different kinds of mechanisms. Or let's say it has 3 attributes that make it a very powerful natural drug. The first is that this end of the molecule binds to the antigen. So that is the immunizing thing, the bacterium or the virus or whatever. So this end here, which I've marked as V, the variable domains, these differ in every antibody, and each one of them can be cognate to a particular antigen. So they can bind this agent, and that act of binding can actually be sufficient to prevent the action of that agent. So for example, you don't need anything more than binding. So let's suppose you've got a toxin present, a snake toxin or whatever. The simple binding of the antibody can prevent that toxin binding to a natural receptor. So in that sense, it's just acting by one of its characteristics. Another characteristic of antibodies is they hang around in the blood for a long time, have a long serum half-life. And this comes from 2 features. The first is its large size. So antibodies are very large molecules, 150,000 in molecular weight. And that means that they escape being filtered by the kidney. Essentially, if the molecule, for example, if we were to take a normal drug molecule, this would go through the kidney very fast, essentially within less than half an hour, probably about 10 minutes. It will just simply come through, will be filtered through. But an antibody escapes that filtration by the kidney because it's too big to go through. But there are other things acting in the serum which mean the antibody has a long half-life. And that is that there is another process going on which is clearing things from the serum, and that's called pinocytosis. And essentially the cells lying in the blood vessels are taking constant little sips all the time of any liquor that's around them, so of any liquor in the serum. It doesn't matter what's in it, they just take it into the cell and they chew it up. And so in fact, if you have, so the natural, let's say, half-life of an antibody in those conditions, if it was subjected to this pinocytotic action, would be about 24 hours. But actually, the cells have a special recycling receptor called FcRn, which any antibody taken in, it captures it and throws it back to the surface. And that can give antibodies half-lives in serum of between 2 weeks and a month. So essentially, that protective agent is hanging around for a long time in the serum because of these two mechanisms. And in fact, the receptor for FcRn is based down at this end of the molecule. So this is the bit that recognizes the antigen. This is the bit that recognizes that receptor. Now, this is the really important end down here, both for this recycling receptor, but also for recruiting killing. And so this end, if the antibody is attached to a virus or bacterium at this end acts like a flag for the immune system. And that leads to the killing of the agent by multiple mechanisms. So what this end does is to flag to a whole variety of immune cells, basically come and get me. There are receptors on neutrophils, on macrophages, on what are called natural killer cells. All of these have slightly different receptors, and the antibody is attracted and will go in and directly kill those cells, for example, by releasing superoxide, or for that matter, conducting phagocytosis, just taking it and chomping it up, or by releasing all kinds of membrane attack complexes against the pathogen. So that's the way in which the antibody works naturally. In fact, however, the money or the interest in antibodies didn't come from making antibodies really against infectious disease. We have those anyway. It came from the possibility that you could use them to treat non-infectious disease. And in particular, the areas where antibodies have been very successful have been in treatment of cancer and in treatment of immune disorders. And this just shows the power of antibodies in two situations. And these were antibodies, both of which I was associated with. So this was a collaboration we had in Cambridge in the late 80s, where we made an antibody that attacked human lymphocytes. So this was actually a collaboration with Herman Waldman, who made that antibody. And this was put into a patient with non-Hodgkin's lymphoma. This is an old lady, and in fact, she's got several kilograms of tumor in the spleen. And after 30 days of antibody treatment, you can see the tumor has gone. And effectively, we're able to give her about a year's worth of extra life as a result of that treatment. Now, antibodies have been particularly useful for treating breast cancer and bowel cancer. You may have heard of Herceptin, which is the anti-breast cancer one, Avastin, which is the anti-bowel cancer antibody. But they've also been used for treating bone cancer and a variety of leukemias. In this case, these antibodies are acting, by and large, by binding to the cell and doing two things. In some cases, blocking the binding of a growth receptor, a ligand to a growth receptor. So, for example, if we think of EGFR, which is epidermal growth factor receptor, then basically when that binds to its natural ligand, that actually helps to trigger the cells to divide. And if you can block that interaction, then the cancer cells won't expand. So they can act simply by blocking, but they also act by their killing action. So they, at the same time, are recruiting a whole series of killer cells, in fact, to clean out the tumor mass. In the case of immune disorders, quite often these antibodies are acting more by simple blocking. So, for example, there's plaque psoriasis is a disease which is associated with high levels of a molecule called TNF, which is an inflammatory mediator. And if you treat this with an antibody which simply binds to TNF and stops TNF binding to the inflammatory receptor, then effectively you can greatly ameliorate these conditions. In fact, the same antibody is used for treating rheumatoid arthritis. So these antibodies have been quite dramatic when applied to non-infectious disease. In fact, they've become so successful. This shows you a chart of pharmaceutical sales for the year 2010. And what you might expect to see here are things like Lipitor. These are the statins. These, for example, Advair is for treatment of asthma. But all the ones, Crestor is another statin, but all the others in the top range are antibodies. So in fact, 6 out of the top 10 therapeutics in 2010 are antibodies. And now Humira, which is that antibody which was directed against psoriasis I showed you and against TNF, is now the world's #1 product and sells $9 billion per year. And all of these things came from engineering proteins. I'd like to sort of try to make a comparison. I've shown you what the features of the antibody are. I'd like to compare that with a classical small molecule drug. So what are the advantages in green that an antibody would have over a small molecule drug? So I've marked the relative advantage in green and the disadvantage in red. If we look down here at the advantages of the small molecule drugs, the beauty of these is that they can get into pockets, they can inhibit enzymes because they get into the active site of the enzyme, they can get out of the blood, they can get into cells. You take them as pills, right? You don't usually have any immunogenicity problems, so if you take them, you won't normally expect to get some rash, or in fact worse. And they're usually made by chemical synthesis, which means they can be made in vast quantities once the chemists get their processes sorted out, which in fact means if it's successful, the companies can make vast profits. Antibodies are very different. So antibodies, essentially, the top of the molecule is big and fat. It doesn't usually get into pockets. They're pretty much stuck in the blood. They get out of the blood with low efficiency. They can't really get into cells. If you try to take them through the gut, essentially they'll be digested because they're proteins. They can be highly immunogenic, and you certainly can't synthesize them chemically. You have to go through very expensive fermentation properties. But they have these other great advantages here because they've got the top of the antibody is a large and extremely complex site which can interact with the antigen, you can develop a very high target binding affinity. By the same token, because it's a very complicated surface, you can get a high target specificity. Chemical drugs are much smaller and in fact therefore tend to have much lower affinities and in fact tend to be much less selective. Quite often they're hydrophobic and will just drop in to other proteins that you don't want them to inhibit. And that's the worry. You go into clinical trials, it looks like it works in your model, in your system, but then you'll discover that actually you're getting something you hadn't expected because they're binding elsewhere in the body and attacking inhibiting some other process. So in fact, chemical drugs tend to have a high off-target toxicity from time to time, whereas antibodies are exquisitely specific, only really going for the site you want. Unlike these chemical drugs, they have a long serum half-life, so you can inject them and then come back a month later and do another injection. And in one case, even 6 months later, because they're so well sustained in the serum. They have all of these killing mechanisms, and you can stop the binding of proteins to their receptor. So these are the advantages of antibodies over the traditional small molecule drug, and they've actually meant they've become bestsellers. So let's look then at the kind of path by which the antibody emerged from its role as a natural defense molecule to a top-grade pharmaceutical product. And I think if I can explain simply, the first step in this was the creation many years ago of what's called the mouse monoclonal antibody, which I've marked in red. Imagine you take humans, you want to kill human cancer. So you take those human cancer cells, you inject them into a mouse. That mouse will make antiserum against those cancer cells. However, that antiserum will attack both the cancer cells and normal cells, because cancer cells look much like a normal cell. They've got a few differences, but by and large, the antiserum that you've made will kill off both cancer cells and normal cells, which actually means it would be toxic to put it into people. But what Cesar Milstein succeeded in doing was making what are called monoclonal antibodies, which were antibodies. He managed to find a way of taking that complex response where you get many different antibodies, both to normal and to cancer components, and sort of resolving it into its components. What he did was essentially take the spleen with all the different antibody-producing cells, fuse them to a cancer cell line, and then he could grow up each of the hybrid cells individually and analyze the properties of the antibody from that. And because you could grow them indefinitely, you could succeed in making antibodies which were which you could just fish out those antibodies directed against the tumor. So that looked great. People talked about them in the 70s as being the magic bullets that were going to kill cancer. The problem was when you put them into patients, they killed they knocked down the tumor, but after about 10 days, you got a terrible immune response to the mouse antibody. So that antibody you put in to do the works basically ended up being seen as foreign, and your own body mounted a response, and in the end, you could actually get terrible immune reactions to it, including death by virtue of the seriousness of the immune reaction. So how to deal with that? We clearly needed to create antibodies that were essentially, they could do all that mouse antibodies could do, but they needed to be human, basically. And so the sort of idea was, could you evolve, actually I couldn't find a mouse version of this, could you evolve a mouse, an animal antibody into a pure human antibody? And in fact the approach we took was to use genetic engineering to do that. Well, in fact genetic engineering, as I said, was the mechanism. And in fact, the antibodies I'd point out is quite a big, complex structure, a horrendous size to engineer, with all these different globules in there. 2 heavy chains, 2 light chains. But actually, at the gene level, each of these domains is arranged quite conveniently as a little bit of DNA in the chromosome. So all of these bits are quite conveniently arranged, and that means that you can consider cutting and pasting them. And so if we look at this, I look just up to this point here where we've got something, we've gone as far as we can go, we now want to go as far as we can go from the mouse antibody. So I'm just stopping short at this point before we come to the genuine human. And essentially the simplest type of antibody was this, where we know all of these things are independent units, and therefore we can just put them together. We know they fold up independently. We knew that already. They essentially are plug-in modules. That's how biology works. A lot of it's evolved to work as a plug-in module. And when you take the section of an antibody that corresponds in a mouse hybridoma that corresponds to your cancer antigen, you can then plug that into a complete human antibody, the rest of the genes of a human antibody, and you end up with an antibody that's about 2/3 human, but has the activity of the mouse antibody. Okay, so I wondered if we could go any further, whether we could go further than that, and if we look at this, the sort of top of the molecule, that section we've just transplanted, essentially you've got about 6 loops built onto a beta sheet scaffold here. And I wasn't very confident about making changes and trying to predict these by any computational method, but it did occur to me that essentially this scaffold looks very similar in structure in a mouse antibody as well as a human. So maybe if we just took the scaffold from a human antibody, then just plugged in the loops from the mouse, we might be able to create an antibody that's much more human. In fact, the point of the scaffold is it's holding those loops in a defined configuration. This provides a relatively rigid surface for the antigen to bind to, which actually, that rigid surface gives you the high affinity, essentially, because you've, if it's highly flexible, it's difficult to get a good binding affinity. Essentially, you're having to, because you're having to immobilize those things and fighting against entropy. So I wonder whether it's possible to transplant those, and the answer is yes, you can. You can transplant them, and in fact, these antibodies can be made to be as essentially as good as the original mouse antibody. You can transplant the activity into a human antibody. So now only about 5% is mouse, and the vast majority is human. So that's actually as far as we could go with that. And now so far that hasn't involved any mathematical principle at all, other than what I call sort of engineering nous and just trying to minimize actually any use of computation if one possibly can. So I'd like to take this stage of trying to create genuine human antibodies. And this does use a minimal amount of mathematics. How would we? They've got particular problems in thinking about creating human antibodies. Because actually, I've told you that antibodies that people are really interested in are the antibodies that are attacking human tumors or human immune components. But actually, if you were to immunize a human with human TNF, they won't make a response to it because it's self. So for any of your own proteins, and that includes proteins on cancer cells, in general, your immune system won't react to it. Essentially, your immune system have all sorts of mechanisms to make sure it does not react to self. It's called tolerant. So how then could we make human antibodies, genuine human antibodies to human proteins? Well, obviously we have to step outside the immune system, and the idea was as follows, that we'd go into human blood and we'd fish out human antibody genes. So let's imagine we have one B cell, which I've marked here, which is producing in black here and gray an antibody which consists of a heavy and light chain variable region. And we've got another B cell in that same population producing a red antibody, the dark and the light red. These two will encode a particular antigen binding specificity, and this will not involve reaction to self. But suppose we were to take those genes and shuffle them, we generate 4 combinations, the 2 original and 2 new ones. So we've got red and we've essentially, when we put red and black together, those are new combinations. And so we wondered if we could generate by essentially using the power of large numbers. So imagine we've got 10 to the 6 B cells. We can then generate 10 to the 12 possible combinations, which is actually more than we can readily screen. So we thought, okay, so we can generate very large numbers, and the vast majority of those will actually be entirely new. So effectively, we've created a vast range of new activities, among which we may find those which are binding to the human antigen. Okay, so how do we find, out of these million different combinations, how do we find the ones we want? And this is where we used a method which had been developed earlier for looking at small peptides, which effectively involves linking genotype and phenotype so as to give you a Darwinian package. So what we put here was the antibody heavy and light chain genes fused to the tip of a bacterial virus. So effectively, you've got the genes here which are linked up to the antibody genes are directly conjugated to the minor coat protein gene of the virus. So if we do that and we clone it and we produce in bacteria, we can take a repertoire of these, so our 10 to the 12 repertoire, we can stick in these phage, so each phage is different. And then when we want to find the one we're interested in, the one that binds to TNF or whatever the antigen is, we can pass those phage down a column and we can pull out the ones we want. Now, in fact, practically, you would never achieve a 1 in the 12, 10 to the 12 enrichment. It's just not going to happen. If you're lucky, you get about 1,000-fold enrichment. But because we've linked genotype and phenotype of these, we can grow them up again. We take the enriched speed, grow it up, and pass it down a column again. And by the iterative cycle, we get another round of enrichment, and therefore a millionfold. And we can just keep repeating this in order to fish out the one we want. So basically, that shows the power of large numbers, generating the large numbers, and then secondly, finding very powerful genetic ways of screening those large numbers. So by using this technology, and this was actually what Cambridge antibody technology was built on, was this approach, we actually managed to make several therapeutic antibodies. So this is actually is the sort of evolutionary path from the early antiserum through to the rodent monoclonals, through to the chimeric, through to the humanized, and then with this conceptual and practical gap, genuine human antibodies. Actually, interesting enough, they all work quite well. The original mouse ones don't work. These are quite good. This shows a timeline from 94 to 2011. And what we can see here, the first class of antibodies was the chimeric. The biggest class is the humanized. And then we see a series of human antibodies, which was of later inventions, coming on and producing a whole range of antibodies. So generally, the humanized, there's no evidence that the immune response that's generated to the humanized is any worse than that which is generated to the human. There is some evidence that the chimerics are worse, as perhaps you might expect. So then I wanted just to simply discuss briefly what next. So we've got this very interesting thing where I described this. We've suddenly found in the last 15, perhaps 20 years, an entirely new pharmaceutical class of drugs that has come on and taken the whole field over. And it's come from basic research, and in fact it's come from, although I've described it as though we had a problem to solve, I'm not going to go into why we actually decided to do those experiments, but it was nothing to do with solving a problem. It was just following our curiosity because we wanted to see what would happen. So what one can think now is to say, well, what next? Is there something else that one might do with this type of technology? So let's just go back to the antibodies and the chemicals, advantages, disadvantages. Wouldn't it be great if we could make something only had advantages and no disadvantages? So we thought, yeah, okay, let's do that. So let's go back to the key feature of the antibody, which is we've got this scaffold with all of these loops on top. And the loops are being held there nicely in position. But in our earlier work with antibodies, we discovered that on the whole, you don't always need all of those loops. In fact, sometimes you can get away with just 2 of the loops. And you've got all of this sequence here, all of the structure, which actually is unnecessary from the point of view of binding. It's just holding these loops in the right place. Now, if we look at this, if we go back to this class here, if we think of the disadvantages of antibodies, several of them are simply due to the fact that the antibodies are rather large. So, for example, it doesn't get out of the vasculature, out of the blood, because it's too big to go through the capillaries. It doesn't bind to pockets because it's too big. Actually, we can't chemically synthesize it because it's actually be too big to chemically synthesize. So we thought part of the solution may be, given that we know that we can get antibodies to work with just 2 loops, could we shrink it down to 2 loops and then find a way of dispensing with all of this rubbish here, all of this unnecessary sequence, because all it's doing is holding things. And we came across a paper by a Dutch group where they had taken essentially a peptide with 3 cysteines on it, marked in yellow, and they'd conjugated this to a benzylic halide in water, and this works beautifully. And you end up then with essentially 2 loops, which are nicely anchored in the middle with this group. And so this group is taking the place, in our minds at least, of all this beta sheet. So we thought, okay, well, how would we make these of any activity we want? So we've got the format right. That's the kind of structure we'll use. How would we if we wanted to make one of these against TNF, for example, how would we do it? Actually, that's just to point out, so far, we've solved the first problem. This thing is small enough to do by chemical synthesis. So that's just, we've got all these boxes here to fill up. So the idea of trying to make those with high affinity, we thought, let's go back to that very powerful, the power of numbers, the combinatorial power. Maybe what we could do is take a peptide and make random sequence here in what will be loop 1, random sequence in loop 2, etc. And so we'd take a peptide, display it on phage, and we'd have cysteines located at these positions. Then we'd add this trisbromethylbenzene to the phage and hope it didn't kill the phage off. And then we could select this entity on our target protein, whatever that is, in the same way as we did with antibodies. The problem was it completely killed our phage off. When we added it, it cross-linked to all sorts of things. And we had 2 years of fiddling around with exact conditions to get it to work. But in the end, we found them. And so we managed to get this to work under very carefully controlled conditions to do a spot of chemistry on this living organism, which is the phage. Now we can then go through this whole cycle of selection, and we can make things with powerful, very high binding affinities. And this just summarizes we can make things with high target affinity, high target specificity. We can get them to bind into pockets because they're small enough. And I haven't got time to show you all the data, but essentially that's what we've actually done. I mentioned to you a key aspect of the high affinity was the complementarity, and in fact, a colleague of mine, Christian Heines, who's now in the EPFL in Switzerland, managed to solve the structure of one of these on the surface of a UPA, which is urokinase-like plasminogen activator. It's a protease, and you can see it's fitting in absolutely beautifully into a surface pocket on this protein. So we can make these things beautifully specific and complementary. I also mentioned the fact that the loops, the beta sheet in the case of the antibody is put there to hold those loops in a reasonably rigid configuration. So one question we had is, well, we've got the high binding affinities, but how rigid is this thing? And I'm not going to go into this in detail, but we thought we'd better try and find out. So this shows what we call a bicyclic peptide here. You can try to work out how flexible that these two loops are with respect to each other by putting an electron donor group here and a reporter group here. So this is a dye which can be quenched by electron transfer from the tryptophan, but only if the tryptophan is in close contact. So actually, if you take a linear peptide and you put those two on in these two positions here, they will actually come together quite frequently, and using fluorescence correlation spectroscopy, you can see quite a rapid time constant for that. So in the order of about 100 nanoseconds in this case. If, however, we put this in, we now find that we're into the 10 microsecond range, which tells us that these two things are not coming together, as in the linear, probably about 100 times more rigid. So that does actually suggest that certainly compared to a linear peptide, we're actually much more rigid. What I know is we're not as stable as an antibody. We're not as rigid as an antibody, and therefore, in fact, it'll end up being more difficult getting the kind of very tight binding affinity you can get with an antibody. So what I've put here then is my, and I don't have time to go through all of these because we're running a bit late, but my realistic prospect of where we can be. I think we can sort most of these out. I think we're never going to be able to get the same very long serum half-life as an antibody. There are ways of getting quite good serum half-life, so what we can do is we can attach to our little bicyclic peptide a palmitic acid residue. This binds to human serum albumin, and that bulks it up, so it gives you a half-life extension strategy. So it gets carried around the blood by the serum albumin, and therefore doesn't flush out through the kidneys. And that instead of giving it a half-life of about 10 minutes, extends the half-life to about 8 hours, and we're looking for further possibilities for extension for that. So I think we can certainly get it to be a reasonable half-life. I think in terms of blocking protein-protein interactions, I think it's I think we know we can block them, but I doubt it will work as well as with an antibody because essentially we really need some element of some sort of pocket, even a shallow pocket, for this thing to fit in and be stabilized there. In the current format, I doubt we can get at intracellular targets or get it through the gut, but I don't think it's impossible. And there's a lot of interest from venture capitalists and small biotech companies, particularly in the States, for doing this. But you're really going to need quite a lot of money to develop this very challenging, technically very challenging things. But this whole approach of really shrinking a protein and bring it right down to something that's extremely constrained, practically, I think is going to be important. But also, actually, it may make your folding problem a bit more tractable if you want to look at it that way, because these constrained peptides probably are more susceptible to solution. And finally, if we think about these peptides, although I've sort of said maybe we can have advantages over all of them, over both chemicals and antibodies, the chances are it's going to fit somewhere in between. There's going to be a niche, a clinical niche, where it's going to be particularly useful. And I would say the two areas I think are most interesting are topicals, where we could apply it at very high concentrations to the skin or to the lung. So effectively, we don't have to worry about the serum half-life then, because essentially we can chemically synthesize large amounts and put it on an ointment or an aerosol nebulizer. Maybe we should even link it up to those little bits of DNA and target them there into the lung. And the other possibility is linking cytotoxic drugs or radioisotopes to it. So at the moment, if you take, people have attached, to give antibodies more killing power, you can attach cytotoxic drugs to them. And, but there are a lot of problems in doing that. It's a problem in getting the you have to do two things. You have to ferment the antibody, and then you've got to do chemistry on that fermented product. And you've got to get it absolutely specific in the place you want. You've got to conjugate that cytotoxic drug onto the antibody. That's very difficult, and technically, yields are low, and it also is surrounded by a patent minefield. So all the players are very well established and very difficult to get in there. One of the things that we might have in our favor with these peptide bicycles is we can just synthesize them directly with the chemical drug on. We don't have to go, everything can be done by chemical synthesis, so we don't have to bother about fermentation. The other feature of antibodies, for example, for imaging studies, if you've got a tumor, antibodies you can show that if you want to image people, you can identify with radioactive antibodies where, for example, the tumor is, by comparing PET and CT scans, etc., and gamma imaging. But the problem is that usually you have to wait a long time for the antibody to be washed out. The antibody tends to go in, hang around the serum for several weeks, and you really can't get the background down off the tumor. So in fact, it's been shown that it's much better to use much smaller molecules which clear much more quickly. So effectively, you give someone a radioisotope and then take a picture of them, and that they wouldn't have to go home in the bus laden with radioactivity, because they'd have pissed most of it out by the time if you keep them in hospital for the day. So I think that there's a considerable interest in having things with actually a somewhat shorter half-life than the traditional antibodies, but nevertheless could target tumor masses. So, I mean, that really I kind of hope would sort of give you a flavor of how far we can go with pretty minimal mathematics. So actually, if you can imagine applying some great mathematics, no wonder, just the sky's the limit as to what one could do. But you have to think about what are you going to apply it to? What's the problem? Because many practical problems can be solved by relatively simple, carpenter type, which I consider myself as a protein carpenter. Nothing sophisticated. Chop bits off and join them up again. You can solve a lot of problems that way, because that's actually the way that nature's done it. It's done these primitive operations, swapping bits of DNA over, and then actually selecting the ones that work. So, thank you very much.
We have run a little over time, and the drinks are waiting outside, so I think we should call it a day and continue discussing perhaps over drinks. So I'd like to thank all the speakers. I think we've had a wonderful feast of lectures from the beginning. So thanks to the speakers from before tea, the DNA, and to our speakers on protein folding and protein engineering after tea. And now let's go and enjoy the reception. Thank you all again.